All right, so today we're going to look at special dominance scenarios when we're looking at Mendelian genetics. There's just a few nuances between this and the simple dominance that we've been looking at, so it shouldn't be too terrible for you to get a grasp on. The first we're going to look at is incomplete dominance. In incomplete dominance, what we mean is neither allele completely dominates the other one. As we've seen before with simple dominance, if you have like a big R and a little r, whatever big R and little r stands for, the big R, because it's dominant, and the little r because it's recessive, the big R would cover up the little r. Not so anymore in incomplete dominance. In incomplete dominance, the big R can't completely dominate the little r. And you'll see this notated a couple different ways. Sometimes people will continue to use big R and little r, and that's how we'll do it in this class. Sometimes for incomplete dominance, you'll see things like R and R prime. But we're not going to mess with that. We're going to use this, and you'll just have to look at the genotype and phenotype ratios or from the directions for the problem to find out if it's incompletely dominant or not. And one way that you can do that is by noting that a heterozygous individual, so in this case big R, little r, they would have a blending of the two traits that are represented by those alleles. So if big R represents one thing, little r is another, if you get one of each, they would blend those two together. So here's an example. If this is probably the most famous example. If we look at snapdragon flowers here, homozygous little r codes for white snapdragon flowers, whereas big R gives you red snapdragon flowers. Now, when you do the Punnett square for this, and let me just mark it out real quick here. So big R with a little r, you can see that all of the offspring are going to be heterozygous. And because this is incompletely dominant, the red and the white blend. Well, think about with paints. If you blend red and white paint together, you're going to get pink. So when a red and a white snapdragon breed, all of the offspring, all of them will be pink. Now, when you take two pink ones and you breed them together, you can get the outcome here, which you can take a look at if you were to do a Punnett square with two pink flowers. So I'll let you do that on your own. Or if you do a pink and a white, you get some interesting combinations as well. So this is what's going on in incomplete dominance. Now this is just one example, but anytime you have incomplete dominance, the two phenotypes blend together. If you take two of the blended ones and breed them, then you get about a fourth of one type, a fourth of the other type, and then about half of them being the blended one again. So that's incomplete dominance. Another example of special dominance is codominance. Now think about what the prefix co means. Like if you have co-captains on a team, that means they're both captains. Okay? It's not like you blend the two together like we blend in incomplete dominance. And again, that's a key word for incomplete is blending. In codominance, both of the alleles contribute to the phenotype of the offspring. So in other words, if and in codominance, we always use two capital letters, so maybe a big B and a big W. These both, whatever they are going to show up, but they don't blend. They appear separately from each other. So obviously, if you think about this, there's some traits that may not work for this. For example, in incomplete dominance, you could very easily look at, say, if big T is tall and little t is short, think about what an individual would be if they're heterozygous. Okay, if this is incomplete dominance, those are going to blend. So this would be medium height. In codominance, because they show up independently, would height work? I mean, it's an oxymoron to say that somebody is tall and short at the same time. You don't have a tall, short person. So in codominance, there's no blending. Both traits appear separately. And so because of that, there's obviously some traits that that's not going to work for. But let's look at an example of one that it does work for. In chickens, feather color is codominant. So we've got two alleles here. B obviously stands for black. W stands for white. And right here, this chicken has a big B and a big W. Notice they're both capital. They're both dominant alleles. Neither one can cover up the other one. And so they show up independently. If you look at this picture really closely, let me make it a little bigger here. Notice that the feathers on this chicken are either black or white. They do not blend. They both show up independently. So if we blend a speckled chicken, that's what we call that here, with another speckled chicken, here's the Punnett square. Think about what big B would be. Big W, and then think big B, big W. Well, if we look at their offspring, some of the offspring will be black. That'd be the big B, big B. If we look at big W, some of the offspring would be white. So about 1 out of 4. Obviously, then the big B, big W are going to be the speckled chickens, just like the parents. So this is an example of codominance because when you have a heterozygous individual, both traits show up separately. And so hopefully you're noticing that with incomplete and codominance, we really are only affecting the heterozygous individual. All right, so the last type of special dominance is called multiple alleles. So multiple alleles, it means that there's more than 2 different alleles that exist. Now, remember, each person or individual can only get 2 alleles for a trait, one from mom and one from dad. If you're not sure why that happened, go back and review the meiosis video. And some alleles are more dominant than others. But like I said, each individual still only gets 2 alleles, even though, if there could be 4 in the population, depending on the trait and depending on what species we're talking about, there could be several, like up to 10 different alleles for a trait, but each individual still only gets 2, one from mom and one from dad. So probably the best example of this would be in another creature here, in rabbits. So in rabbits, we have, let me grab my tool here. Oh, it disappeared on me. Hold on. So in rabbits, fully colored coat, that's what we normally see here in Nebraska, kind of this boring brownish grayish rabbit. That is represented by the allele big C. A rabbit with a gray coat or chinchilla is C with a superscript CH. Now keep in mind, this is only one allele right here. I know you're thinking, Mr. Seggerman, there's 3 letters there, but that C, CH is only one allele. Then we've got Himalayan rabbits. That's this one right here. That's coded for by C with the superscript H. These rabbits, it's interesting, this allele, what it does is by default, the rabbit's white, but if a particular area of the rabbit gets cold, it actually turns dark. And think about the areas where it turns dark. The ears, the nose, the feet, and the tail. Think about where the Himalayan rabbits live. The Himalayas, it's really cold there. So think about why they might want to die, or not die, but change the color of their fur to black on these outlying areas of the body. Think about when you go outside, what's the first parts of your body to get cold? That might be a hint for you. And then the last kind here is the albino, and that's represented by a little c. Now this order of dominance is important. This is the most dominant, and this is the least dominant. So you can imagine, we can have any combination of 2 of these alleles. So for example, big C, well, that'd give us a normal colored coat, but so would big C, CH, or big C, CH, or big C, little c. All of those would give us a normal colored rabbit because this dominates everything below it. Same thing with chinchilla. If we had C, CH, C, CH, that'd be Himalayan. Or if we had C, CH, CH, that would also be, that'd be chinchilla because this covers up anything below it. All right, so we're going to look at an example that exemplifies both multiple alleles and codominance. And to do that, we're going to look at blood types. So there's 2 words that you need to know about with blood types, and they're called antigens and antibodies. Antigens are like name tags. So if you have a red blood cell, so let's just draw a little, save cool properties. So if we have a red blood cell here, it has these chemicals on them called antigens, which basically act like a name tag. They kind of identify what the blood is. So, if it's type A, it's going to have A antigens on its blood that tell its body, hey, I'm an A blood cell. Now, each person only has one type. So, if you're type A, all your red blood cells will have A on them. Antibodies are like hitmen. So their job is to take out specific cells. And so people who are not type A blood, so for example, if they're type B blood, they actually have antibodies that are A antibodies, that their job is to hunt out and kind of attack any red blood cells that are type A. So you can imagine that a person who's type A blood would not want to have A antibodies because that would obviously clump their own blood, so that'd be bad. So if we look at the different blood groups here, we have four types. We have A, B, AB, and O. So again, the antigen present on A blood is this A antigen. So there it is. The group B blood, they have the B antigen, this little name tag that says, hello, I'm type B. AB blood has both A and B antigens. So it's like multiple personalities. It says, hello, I'm type A and B. And then group O has no antigens. It doesn't know who it is. This antibody part is also very, very important. So if we look at group A, they are an A blood because they have A antigens on their red blood cells. Notice what kind of antibodies they have. They have anti-B. Think about that. If they would have anti-A, it would attack their own blood. So they don't have that. But they don't want to have any blood cells that say have a B antigen on them. So in a person who's type A blood, they have anti-B antibodies, which, if you got a blood transfusion that had B blood in it, these would attack it. That could be very bad. It could actually end up killing you. A person with B blood, again, they have B antigens, but they have anti-A antibodies. Person with type AB blood has both, so they have no antibodies for blood type. They have antibodies against bacteria and viruses and whatnot, but not for blood type. Think about why. If they had anti-A or anti-Bs, it would attack their own blood cells. And then finally, type O, they have no antigens on their blood, but they have both A and B antibodies. Sorry for that little message there. So moving on here. Let's just kind of fill out this little table. So the allele symbols, these are what we actually use when we're doing a Punnett square. The first one we have is IA. That's represented by the letter I. So IA is the allele that causes you to have A antigens on your red blood cells. So that would, contribute to the blood type A. Again, if you have type A blood, you're going to have anti-B antibodies. The next allele is IB. Again, I'm rewriting it because it's capital I. That contributes to the B blood type, which would give you B antigens on your red blood cells and anti-A antibodies. The last one is little I, and that means that you're not going to have any antigens on your red blood cells. That'd be type O blood, and so you'd end up with both A and B antibodies. The only one that's not on here is if we have type AB blood. Think about it. If IA causes A antigens and IB causes B antigens, type AB blood has to have both alleles. So if we go back to this diagram up here for these blood groups, group A, they could be IA, that would be one genotype that would give you A blood, or they could be IA, little I. Because think about that. That little I means it's recessive. Group B could be IB, or IB, little I. Group AB, as we already said, has to be IA, IB. And group O, again, since that's recessive, they have to have two recessive alleles for group O. All right, so here this is just kind of spelling that out again. It also gives you some hints about who can receive from whom. But that's not the only way that we can classify blood. We also have this thing called Rh factor. Now, don't get confused with this. This is a different gene altogether. So Rh factor has no bearing on if you're A, B, AB, or O. But a lot of times you'll hear something like A positive blood. Well, that just means that they're type A blood, so they have A antigens on their surface. So if this was a red blood cell, they'd have little A's on them. The positive means they have this thing called Rh factor. So in addition to those A antigens, they would also have Rh antigens. So an A positive person has A antigens for blood group, and they have Rh antigens for this Rh factor. So the alleles that we use are Rh plus. So the allele Rh plus says that you're going to have Rh factor on your blood cells. So again, if you are a what we'd consider positive blood type, the genotypes you could get would be Rh positive, or you could be Rh positive, Rh negative. So what does that tell you about Rh positive allele? It's dominant. The Rh negative allele is recessive. So somebody who has the Rh negative allele, that wants you to have no Rh factor on your blood. So a person with this, if they're what we call negative blood, that means their genotype has to be Rh negative, Rh negative. So if we look down here at this table, allele symbol Rh positive, that gives you the Rh antigen and no Rh antibody. So that'd be a positive. And it doesn't matter. You could be A positive, B positive, AB positive, or O positive. The Rh minus allele gives you negative blood, but you have to get two of them, remember, because that's recessive. And so you do not have the Rh factor on your blood, but you would have, and this is important down here because this is a key thing when we're doing blood typing, you would have antibodies against Rh. So that has some implications when we start donating blood. So this little chart here, I had students fill this out for homework, but this little chart, oh, I lost my little tool again. Hold on. I don't know why it keeps disappearing on me. It doesn't like me today. So this little chart helps us kind of figure out who can give to whom. So if this is the donor up here, what this is saying is O negative. Think about what they had on their blood. If you're not sure, go back to it. They have no antigens. So they're kind of like the ninja blood. A person with AB positive has no antibodies for blood grouping or Rh factor, but somebody who's O negative, they have A, B, and Rh antibodies. So they can only get from somebody else who has O negative, no antigens on their surface. So for example, O negative, because it can give to anybody, is called the universal donor. AB positive, think about AB positive. So that means it must have A antigens, B antigens, and Rh factor antigens. And again, I'm talking antigens now, not antibodies. So if they have all these antigens, they can't have any of those antibodies. They can't have A antibodies, B antibodies, or Rh antibodies. Otherwise, it would attack their own blood. So they can get blood from anybody. So AB positive is the universal recipient. So there's some implications here when you're trying to figure out who can give to whom. You have to take into account not only their blood group, but also this positive and negative. There's some cool things here that we can do with figuring out who the father might be of a child. So like if a mother's type A, father's type A, the child could be blood type A or O. So that'd be something that you'd want to go back and look at the alleles for how that could happen. We're going to skip this for right now, and we'll look at a few examples of complex inheritance patterns. So an example would be in cats, black fur is caused by the genotype big B, big B. Oh, that's what does it when I click on that thing. Then I lose my toolbar. Oops. Hello. So black fur is caused by big B, tan fur by big T, and tabby colored black and tan by big B. So think about that. There's two dominants there. And when we get one of each in an individual, we see both black and tan. There's no blending here. So what inheritance pattern is this? Well, since there's no blending, this is going to be codominance. So what percent of the kittens would be tan if you crossed a tabby mother with a black father? So in order to do that, we have to, we're going to do a Punnett square here. So in order to do this, we're going to, what was it again? Okay, tabby mother with a black father. Well, in order for mom to be tabby, she has to be big B, big T. A black father has to be big B, big B. So if we do the Punnett square, we're going to get big B, big B, big B, big B, big B, big T, big B, big T. Now the genotype and phenotype ratios we should be good at by now, but big B is going to be 50%. Big B, big T is going to be 50%. This, so if we do the phenotypes over here, big B is going to be black. And because this is codominant, B and T together gives us the tabby colored. So both black and tan show up separately on the cat. So it'd have splotches of black and splotches of tan. The question was, remember, what percent of the kittens would be tan? None. So the answer to that question is 0%. If we do the what would the phenotypes be of a cross between two tabby cats? Ooh, great one. So let's do that. So two tabby cats. So remember, we have to have one of each to get a tabby cat. So this would be big B, big B, big B, big T, big B, big T, big T, big T. So our genotype ratios, that's going to be 25%. Our big B, big T would be 50%. And our big T, 25%. And again, because this is codominance, when you have this heterozygous, they're going to both show up, and that's the tabby cat. So we're doing our phenotypes now. Big B would be all black, and big T would be tan. Again, not too terribly difficult. If we go to another example here, in humans, wavy hair results from being heterozygous for straight hair, little q, and curly hair, big Q. So the first question is, what inheritance pattern is this? Well, if we think about this, we can have straight hair, or we can have curly hair, or we can have this kind of wavy hair, which is somewhere in between. This is really kind of a blending of this and this. So blending, remember, that's going to be incomplete dominance. So cross a wavy-haired mother with a wavy-haired father. Well, in order to have wavy hair, they have to be heterozygous. So that means that we're going to have a big Q, little q, with a big Q, little q. So we're going to get something that looks kind of like this for our Punnett square. Again, I'm doing it without the lines just to save time, but if you really like the lines, go ahead and draw them in. So if we do our genotypes and phenotype ratios, it's going to be 1 out of 4. And notice how I switch back and forth between using fractions and percentages. It really doesn't matter because they mean the same thing. Big Q is going to be 2 out of 4. And little q is 1 out of 4. So if we look at what these would be then, big Q, again, if we go back to what our alleles mean, that's curly hair. Little q would be straight. And this would be wavy. Again, our ratios look similar to simple inheritance pattern for the genotypes, but when we have a heterozygous individual, they blend. All right, we won't do the last one. We're gonna move on here to our last type of inheritance, which is multiple alleles. So as you can see here, for fur color, there's a lot going on right now. There's 6 different alleles right here. C big B for black fur, C little b for brown fur, little c ch for chinchilla fur, little c d for white with dark eyes, little c superscript b for pale gray, and little c for albino. Remember, this is listed from most to least dominant. So for example, this right here is more dominant than everything else. This is more dominant than everything below it. So there's some implications you have to think about when you're doing your Punnett squares. So what genotypes could give you a dog with brown fur? Well, that'd be any combination of big C, lowercase b, with anything equal to or less than it. So big C, lowercase b, remember that's an allele, that's an allele. That would give you brown fur. You can do big C, little b, little c, ch. That would give you brown fur. So would this. Oops, let me undo that one. That would give you brown fur because this is less dominant than that. So here we're kind of back to simple dominant recessive. Anything that's more dominant covers up the other one. Cross a purebred white dog with black eyes with a dog heterozygous for chinchilla and pale gray coat. Okay, so this gets a little confusing. Cross a purebred, remember that means homozygous, white dog with black eyes. So that's gonna be here, this CD. So let's go ahead and we'll start this one here. So it's gonna be purebred, so C, D, C, D. We're gonna breed that with a dog heterozygous for chinchilla, so that's this one, and pale gray coat, this one. So they're going to be C, CH, and C little b. C little b. All right, so we've got our Punnett square set up, so let's fill this out. C, D, C, C, H. Notice I put this one first because it's more dominant. If we go back to our sheet, the CD, oh, here Mr. Seggerman made a mistake. We're going to put the CCH first because it's more dominant. Oh, come on now. Okay, so then down here, if we look, CB is less dominant than CD, so we'll put CD first. That just kind of helps us keep track of it. All right, so if we do our genotype ratios, we've got 2 that are CCH, C little d, and we've got 2 that are C, D, C, B. So now we have to figure out what these actually look like. Well, CCH, C little d, this chinchilla fur is more dominant to white with dark eyes. So this is going to be the chinchilla. That sounds kind of funny to think of a chinchilla dog, but it's just this kind of a grayish combination of fur. And then we've got the other one, sorry, was C, D, C, little b. So if we look at our scale here, C, D is more dominant over C, little b. So these last 2 dogs are going to be white with dark eyes, as opposed to, completely albino. All right, so there you have it. If you have any questions, feel free to email me or get on Angel or use your textbook.